Dear colleagues, I am Filippo Crea, Editor-in-Chief of the European Heart Journal. Welcome to our dialogue. I remind you that we have a dialogue each month with leaders in cardiology from the 5 continents. The dialogues are Socratic conversations among journal editors, authors, and readers. They are free and live streamed. Thus, please send us your questions and we will answer. Well, this dialogue will have a new format. Our protagonist today is a trial, the VERTIS CV trial, published in the New England Journal of Medicine. But I promise you that it will be an interesting dialogue. Indeed, this important trial will be discussed by Chris Cannon, the first author of the study, by Carlo Patrono, Associate Editor of the European Heart Journal, who wrote a comment on this study in our weekly journal Scan, and John Jarcho, Deputy Editor of the New England Journal of Medicine. Thus today, you will have the privilege of 3 different perspectives, from the author, from the editorialist, and from the editor. I'm sure that you will enjoy it. And now it's my privilege to ask Carlo Patrono to introduce Chris Cannon. Please, Carlo.
Thank you, Filippo. It's a great personal pleasure to introduce our guest author. Dr. Cannon is a professor of medicine at Harvard Medical School and senior physician in the preventive cardiology section of the cardiovascular division at Brigham and Women's Hospital in Boston. He currently serves as the education director in the cardiovascular innovation group. He has worked for 25 years as an investigator in the TIMI study group, and he has been a principal investigator of more than 20 fundamental clinical trials. And just to name a few, Tactics, TIMI 18, Prove It, Improve It, and Redual PCI. And he is the lead investigator for VERTIS CV. So it's a special pleasure to have you with us, Chris. And to start our dialogue, would you like to summarize the study design and main findings of the VERTIS trial?
Well, certainly, and as I say, it's a great pleasure to join you. And I should note that my father has followed and worked with you for so many years, and I follow in his footsteps. So it's a delight to join you and everyone on this call. So the VERTIS CV is one of the cardiovascular outcomes trials of the very exciting class, the SGLT2 inhibitor class. There are now 10 trials. This was one of them. And it was one of the 4 done in patients with cardiovascular disease or vascular disease. In specific, it was patients with prior known vascular disease. And so there are 4 trials, one for each of the currently approved SGLT2 inhibitors. Subsequently, there have been trials done in patients with heart failure and then also with chronic kidney disease. And so this was one of these 10 trials overall. We enrolled about 8,000 patients with prior ASCVD to one of 2 doses of the agent ertugliflozin, 5 and 15 milligrams, versus placebo on top of their usual treatment for diabetes. I should say everyone did have type 2 diabetes in addition to the atherosclerotic cardiovascular disease. And so this was designed to be a non-inferiority safety study as the FDA had mandated, and so did find that for the major adverse cardiac events, the hazard ratio was slightly less than 1, and so did show the safety issue that FDA had called upon. Then looking for superiority on MACE, it was not a significant difference, about 8% lower, but not significant. And then the secondary endpoint that we looked at was cardiovascular death or hospitalization for heart failure, and that tended to be lower, but again, not a significant reduction. The endpoint of then renal dysfunction or progression of renal disease by the first definition we had pre-specified of doubling of creatinine or cardiovascular death or I forget what the other component was, or end-stage kidney disease, that had just a trend of 19% lower. Subsequent analyses that we presented using the definition that all the other trials had used showed the same thing the other trials had seen was a 40% reduction in progression of renal dysfunction. So that was sort of the overall summary. I think in the population was very similar to EMPA-REG, and so those two trials were put next to each other and said, well, wait a minute, there was no difference in mortality as was seen in EMPA-REG. And so we've subsequently also done a meta-analysis of the trials available at the time, and largely find the data are consistent for each of the different endpoints with the other trials, but a lot of our p-values were not significant. So that's the brief overview summary of the trial.
Thank you, Chris. As you will know, many successful drug classes are characterized by large numbers of members, 5 to 20, sharing one or more class effects, reflecting a common main drug target. And examples are NSAIDs, statins, ACE inhibitors, ARBs, and more recently SGLT2 inhibitors and GLP-1 receptor agonists. However, individual members of the same class may have a somewhat different spectrum of efficacy and safety. And these may depend on genetic polymorphisms affecting drug metabolism. For example, in the thienopyridine class, if we compare clopidogrel versus prasugrel, or multiple isoforms of the drug target, for example, COX-1 and COX-2. Also, individual members of the same drug class may have off-target effects not shared by the whole class. So is there evidence of heterogeneity in the cardiovascular and renal benefits of SGLT2 inhibitors? And is the extent of SGLT2 selectivity of any relevance to such heterogeneity?
Well, an excellent question. And this became, as John oversaw the writing of this was a whole paragraph that was, I think, the second paragraph of the discussion of, how do we understand these results in the context of other trials, and are there differences? So, the broad answer is usually a class effect is the answer, so that a given class will have, the overall average effect, and then any trial will have some variability around the mean. But the alternate, idea is, are there actual differences in the drug? And so ertugliflozin turns out to be have identical characteristics to empagliflozin. So in its specificity, I think it's 2,000 to 1 of SGLT2 to SGLT1. And the binding characteristics and the PK and sort of every metric in the actual drug is almost identical to EMPA, and greater specificity than dapagliflozin or other SGLT2. So not much difference pharmacologically. Then the pharmacodynamic effects or the pharmacologic effects, I guess, on hemoglobin A1C, weight reduction, blood pressure reduction also were basically identical to all the other SGLT2 inhibitor trials in this population. So about a 0.5% reduction in hemoglobin A1C, about 3 or 4 pounds in body weight loss, and 2 or 3 points reduction on average of blood pressure. So very similar effects there. So then for the cardiovascular outcomes, one really has to turn to a meta-analysis of the trials and then ask the question, is there heterogeneity? And so that was done. Darren McGuire led this analysis. It was published in JAMA Cardiology. And so the I-square statistic looks at heterogeneity of the effect amongst them. And so most of the endpoints had marked homogeneity. I think for cardiovascular death, there was a moderate lack of heterogeneity, with EMPA-REG being different from all the other trials. For heart failure, it was no heterogeneity at all, with about a 30% reduction. And even cardiovascular death or heart failure also didn't have significant heterogeneity. For the renal endpoint, also statistically, there was some heterogeneity, but we had different endpoints. So using, and we presented at the EASD, the sustained 40% fall in eGFR or end-stage kidney disease or dialysis as the endpoint, and that had no heterogeneity, about a 40% reduction with all the different SGLT2 inhibitors. So I think I came away with the statisticians, or what statisticians have been telling us as clinicians every time we do a trial, that they say, don't look at the p-values, look at the confidence limits. There's variability in that, and that the p-values in the VERTIS trial, for several of the endpoints were not significant, but when you look for actual heterogeneity of the effect, that it falls within the range of what the other trials have done. So I think the variability around the mean of a class effect is what I've taken away, and I think other members of the steering committee as well.
Thank you, Chris. So as you already mentioned, the original sample size of VERTIS CV was intended to assess non-inferiority versus placebo. And this was met by the study results. However, the sample size was doubled halfway through to more than 8,000 patients to include efficacy objectives for superiority with respect to cardiovascular and renal outcomes. So what do you think is the reason that VERTIS CV failed to show superiority on MACE, the primary outcome, as well as on death from cardiovascular causes or hospitalization for heart failure, which was your first key secondary outcome?
2 points of slight clarification. So for the doubling of the sample size, that was done in order for us to get the trial done before everyone was going to be started on SGLT2. So the original design was to do a 4,000 patient trial with 5 years of follow-up, so to have the long-term follow-up. But we realized that trial after trial was positive, that people were not going to allow patients to continue in this population in a placebo-controlled trial. So we said, we have to hurry up before it's too late. So that's why we doubled the sample size to 8,000 and then planned a shorter follow-up. It was about 3 years overall. So then for the testing, we looked for non-inferiority for MACE, but then the actual superiority testing was only planned to be on the CV death and hospitalization for heart failure. And that was not significant, so that then stopped the hierarchical testing officially. We thought that we would see a difference there. I think in the end, the absolute difference was about a 1% difference in CV death or hospitalization for heart failure, and the heart failure was different. But the confidence interval of what the hazard ratio saw, crossed 1, and so it was, I think 0.08 was the p-value, or 0.11, one of the two. So there, I think, again, the confidence limits, simply we got unlucky in demonstrating on that particular endpoint.
Okay, thank you, Chris. Now back to Filippo.
Well, let me introduce now John Jarcho. He is one of the most influential and decisive men in the world in regard to cardiovascular medicine. He is indeed the deputy editor of the New England Journal of Medicine for Cardiovascular Medicine. He's also assistant professor of medicine at Harvard Medical School. John received his medical degree from the University of Utah, and then he started his professional career initially focused on basic research and led to the first identification of a genetic locus for hypertrophic cardiomyopathy. And then in 2002, his professional focus shifted and became deputy editor of Up to Date for Cardiology. Finally, in 2005, he joined the editorial staff of the New England Journal of Medicine. And as deputy editor, he has led efforts to establish rapid reviews and rapid online publication of practice-changing clinical trials. John, welcome to our dialogue. And I would like to start with the first question. What is your attitude towards neutral trials? They are less attractive than positive trials and less likely to be cited, but not publishing them can generate a bias in favor of positive trial. What is your attitude and the attitude of the New England Journal of Medicine?
Well, thank you very much, Filippo, for that perhaps excessive introduction. I'm grateful for your kind words. Let me say, first of all, also what a privilege it is for me to participate in this dialogue session with you, Chris and Carlo. As you know, usually editors are kept silent. It isn't often that we're actually asked to comment on our work, but the job of an editor is, to me, a truly exciting and an important one. It's been a privilege to be involved with trials like VERTIS CV and to play a role in the presentation of this important kind of research to the medical public. So you ask a good question about neutral trials. And I think you're indicating here, journals in the past have been criticized for only wanting to publish trials that showed, enormous positive effects, and for being less interested in other words, to evaluate the trial in terms of its interest based on whether we like the outcome or not, which is not a really very good way to assess the importance of a piece of research. And I think the New England Journal in the past, like many other journals, had been guilty of this sort of biased perspective on what to publish. I do believe that in recent years, we made much more of an effort to publish such trials. And in fact, my counterpart in oncology, who handles the oncology trials of the New England Journal, when I bring a paper, very often says, another negative trial in cardiology. You have nothing but negative trials in cardiology. To which I respond, yes, but you have positive trials in oncology, but the survival difference is only 15 minutes. So I feel that I have some kind of riposte, at least, to his comments. With regard to one of the important developments in recent years, of course, has been the increasing importance of non-inferiority trials like VERTIS CV. And obviously, non-inferiority trials have to be distinguished from what we might call negative trials, that is, trials in which the goal was to show superiority as the primary objective that fell short. Non-inferiority trials, of course, don't seek to demonstrate superiority as their primary goal. With non-inferiority trials, a crucial question that the editors have to ask is, what do you mean by non-inferiority? Because the key with a non-inferiority trial is that each trial has its own definition based on setting a boundary for the 95% confidence interval. In the case of VERTIS CV, and indeed of all the other cardiovascular outcomes trials in diabetes, the non-inferiority margin was specified basically by the FDA based on a single standard for all the trials in that space. In any case, with regard to VERTIS CV in particular, when you consider all of the outcomes, we, of course, have considered all of these trials to be of major importance. We were publishing some of the major cardiovascular outcomes trials of other classes of anti-diabetes drugs that had less impressive positive effects. And of course, nobody was expecting what happened in EMPA-REG. Let's just be clear. In 2015, when EMPA-REG came out, people were astonished. And you have to try to recapture in your mind the mood at that time. I remember that it was actually said that year at the European Society of Cardiology meeting, somebody spoke in one of the sessions and argued that although the cardiovascular outcomes trials that the FDA had mandated were very nice and they'd shown non-inferiority for cardiovascular events, that we were never going to learn anything important from these major trials, and they were costing a lot of money and they were taking a long time, and we should stop doing them. And this was, a month before the results of EMPA-REG came out. So people really didn't anticipate that this was going to happen. And if you, I can only imagine what it was like to be on the steering committee of a trial like VERTIS at the time and say to yourself, oh my God, they've totally scrambled the playbook, and we are going to have to go back to the drawing board and take a new approach to how the trial is conducted and designed, and the power and all the rest of it. And this hit every single one of the other SGLT2 inhibitors, except EMPA-REG being the first, at different time points in their trial conduct, and they had to work out what they were going to do. But then the question becomes, all right, look at VERTIS CV and ask yourself, what is the journal thinking when it's trying to make a decision about publication? And it's correct to say that, if you look at in terms of p-values, and if you look in terms of predefined testing strategies, the results of the EMPA-REG trial look more remarkable than actually any of the other drugs in terms of the number of outcomes that showed formal statistical significance. As Chris has been discussing, the big issue is, well, are the differences between the trials, do they really reflect a biological difference in the effect of the drug? Or are we talking about differences that are due to, the random differences that occur in any such situation? And I think that the answer to the question is that we aren't smart enough to know how to judge these things in advance. And let's just say, as a thought experiment, that VERTIS CV were the first, not the most recent of the, well, actually there's now also the sotagliflozin trials, but let's just say that VERTIS CV had been the first of the SGLT2 cardiovascular outcomes trials to be concluded and reported. Would we have, what would we have chosen to do with that paper? And, I think in hindsight, we can say, oh, well, it would have been important to publish the paper, even though it was simply showing non-inferiority, which was the original goal. I wonder whether, if we didn't have EMPA-REG, and if we didn't have the Canvas program, and if we didn't have Credence, and if we didn't have dapagliflozin, whether we would have looked at it and said, okay, but I'm not sure how interesting this is. So the reality is that we are influenced by the totality of the evidence, as we must all be. It is tremendously different what it means, the results of VERTIS CV, knowing the results of all those previous trials. It's a different situation, both with regard to how we as editors would think about a paper, and with regard to how practitioners have to think about what they do as clinicians with the evidence. You cannot look at a single trial in a vacuum when you have a large amount of data. And I think that, Chris's point when you look at the meta-analysis is that it tends to suggest that maybe not for every outcome, but certainly for at least some of the outcomes, there is a lot more reason to believe in the similarity of the results than in the differences, which is part of our reason for thinking, all right, this is important to publish, it's important to get it out there. We, of course, as the editors, don't take a position on what the results of the trial mean. And in fact, we push authors to be circumspect about saying what they think the trial means, because the publication of a major trial at any one time point it's the beginning of the discussion. It's not the end of the discussion about how you interpret the results.
Thank you, John. Thank you, because I think it's fascinating to have the perspective of the editor as well. Now, let me remind the colleagues, the audience, that they can ask questions. Please ask questions and we will answer you. And now, John, for you at the second question, let's change page. A question which I think is in the mind of many colleagues, in particular younger colleagues. And the question is, what increases the probabilities of having a manuscript published in the New England Journal of Medicine? Difficult question.
Well, certainly a question that I get asked very frequently, as you can imagine. I tell people that there are, several specific characteristics that we're always looking for. We obviously want to publish good work, but what do we mean by good work? Well, there are a number of criteria that I certainly have in my mind when I'm thinking about it. The first is really originality. How new is the idea? Is this the first time this question's been studied? If it's not the first time the question has been studied, is it more definitive than other work that's been done in the area? Or is it complementary in a very fundamental way to previous work that's been published in the area? The second thing that I tell people is importance is key. How significant is the issue in medicine? Is this something that's going to be primarily of interest to subspecialists? In which case it may be more appropriate for your journal than for our journal, or is it of general medical importance? Are the findings substantial enough to be what we would call practice changing? And of course, that's a term that gets thrown around a lot. Sometimes not always clear what it means, but it is a way of thinking about what you mean when you say a study is important. A third thing is quality. How well was the study done? Was the design, the best that can be achieved? And we talk not only here about clinical trials, although of course those are important, but well-designed observational studies are also important. Is the evidence strong and convincing? Is there any indication of bias in either the way the study was designed or the way it was interpreted? The fourth thing is the possibility of error. And I will just say that this is the thing that keeps me awake at night. How high is the risk that the conclusions could be wrong? Is the study in some way potentially misleading? Will the reader take away the wrong message? Or is there a possibility of medical practice being actually adversely affected by what the study seems to suggest, but may be inaccurate in suggesting? And then last, but of course, certainly not least, is ethics. Was the study conducted in an ethical way? Was informed consent obtained? If there wasn't informed consent, and there's an exception, what's the reason? And is it valid? Have there appropriate measures been taken to ensure limitation of risk to study subjects? Has privacy been appropriately protected? Are there any reasons to be concerned about fraud or possible scientific misconduct? So those are the kinds of things that I personally, and I think the editors as a group, think a lot about, that help us to make a judgment. Is this a paper that's suitable for publication in the New England Journal?
Well, thank you, John. I think this is very useful for our audience, the wise words you have said. And as this is a very interactive dialogue, do you have questions for Chris Cannon, perhaps about his next paper for the New England?
Well, I can say that we're always interested in what Chris is doing. So I'm curious about what he's doing that he hasn't told us about yet. But I can think of a couple of things in particular that would be useful coming off of the VERTIS CV trial. I guess the first question is, with regard to the issue of a class effect, I always feel when this issue comes up, there are a lot of reasons to ask yourself whether a class effect is likely or whether individual differences between the drugs are likely. In my view, the most definitive way to confidently address the issue of a class effect would be to perform an actual head-to-head randomized trial, let's say between erdagliflozin and empagliflozin. There are reasons why such trials are less practical than the initial trials. In the first place, if differences are small, you'd have to make the numbers very, very large in order to document a difference. And in the second place, of course, you might encounter some lack of enthusiasm by the funders if the funders are the manufacturers of the drugs. So my question for Chris is, do you think that such a trial is possible? And do you think that people could be persuaded to fund it?
No, unmute, please, Chris. Chris,
Amelia has to unmute him.
Yeah, Amelia,
yeah,
please.
Sorry,
unmute.
I'm just looking for him right now. There we go.
Okay,
I apologize. I was trying to be have my Zoom etiquette, and put myself on mute while others are speaking. And anyway, well, I think this would be a wonderful and good question to ask. And the hope is, could we try and do things like this in a pragmatic sense to have patients get one or another agent as they're being seen in routine practice. And these type programs, I think, would be ideal for pragmatic things where you're not doing a direct comparison of all the small AEs and things that you need in a registration trial, but are looking for any big differences. And I suppose in the ISAR React 5 trial was one such recent study that you had published in the New England Journal. I think some sub-studies in the European Heart Journal as well, that is very helpful to have direct comparison. So I would hope that we could do something, and potentially in the pragmatic side. From the broad science, seeing the similarities, is that a key thing in limited resources of, would you want to spend another $500 million to see, is there a slight difference in drug A versus B? That's where you'd like to try and do things like that in a pragmatic sense. And I should point out that there are other areas of variability. And I think one that I've looked to was in the 2 heart failure patient population trials. So DAPA-HF and Emperor reduced. One had a reduction in mortality and one didn't. And in the cardiovascular outcome trials, the reduction in mortality was the opposite. So in the CV outcome trial, it was a significant reduction in CV mortality with EMPA-REG, but in the heart failure, it was with DAPA and not with EMPA. And so they're 2 well-received drugs. Everyone believes they both work terrifically well, and yet stark variability in the hard endpoint of cardiovascular death. And so that was a reminder to me that even with large trials with hard endpoints and proven drugs, there is variability in the point estimate that can be seen in a given trial.
Thank you, Chris. And now back to Carlo. Carlo, I'm sure you have other questions for Chris.
Of course. So Chris, let's go back to the recently published meta-analysis that you just mentioned of the association of SGLT2 inhibitors with cardiovascular and renal outcomes in patients with type 2 diabetes. You emphasize the similarities that you found in this meta-analysis. Let me play the devil's advocate and emphasize the differences, because you did find important differences in outcomes associated with 4 different compounds. So first of all, the beneficial outcome on MACE was rather modest, but was demonstrated only within trials with empagliflozin and canagliflozin. Likewise, only empagliflozin demonstrated significant cardiovascular death risk reduction. And you also acknowledged that there was moderate heterogeneity across the class. And finally, ertugliflozin was the only SGLT2 inhibitor of the 4 studied that failed to reach statistical significance on the pre-specified kidney composite endpoint. So what would be your interpretation of these divergent findings?
Well, I think and I realize I didn't answer your first question on why do I think MACE was not shown. So starting with the MACE endpoint, the overall effect from the meta-analysis is a 10% reduction. That polling the 4 or 5 trials together is significant. And the heterogeneity, as noted, comes from empagliflozin having a larger effect than all the 3 other drugs in the 4 other trials in that one. For the renal endpoint, I think the definition is the key. So we had pre-specified doubling and not sustained doubling, as was the case in EMPA-REG. And then looking at sustained 40% reduction, which had been used in subsequently in EMPA-REG and the other 2, the findings are identical. So there's in all trials a 40 plus or minus 2% reduction in progression of renal disease. So I think there the definition that we had pre-specified being different was why that had a 0.81 hazard ratio as opposed to 0.6 in all the trials and the subsequent analysis. As with a subgroup analysis, if you look at men and women in a given trial and you say, oh, it's only significant in men and not significant in women, that would be one way to say, let's look at the actual findings in that group. But the sort of statistical way is you're supposed to look at the heterogeneity, p-value of her subgroup, or the I-squared heterogeneity across trials. And that's where there's not really much heterogeneity in looking across all the different ones. And then I come back to, well, does it make any sense that there would be a difference of drug A versus B? And we can't come up with any reason other than variability of the confidence limits biologically that there would be.
Thank you, Chris. However, when I read your discussion, the discussion of this meta-analysis, I found of some interest the fact that you emphasize that these discordant observations, while not attributable to differences in the doses studied or the different drugs, may reflect the fact that the cardiovascular and renal benefits of SGLT2 inhibitors are due to mechanisms other than SGLT2 inhibition. So would you like to expand on these potential off-target effects of SGLT2 inhibitors?
Well, now I'm definitely out of my expertise. There are, it's a phenomenon that's really neat, that when a class of drugs works, we then try and figure out how they work, and so I suppose to some degree, aspirin is even in that category of 100 plus years of what degree are things responsible. Certainly statins, how much is related to the LDL versus anti-inflammatory effect. And I think we've come to realize that this class of drugs has many effects. My simpleton approach is that the changes in the kidney and the feedback in the glomerular and efferent and afferent loops is the key source of benefit, in that the pattern looks very similar to MRAs and ACE inhibitors and ARBs. But there are many other mechanisms that are being looked at in the myocardial cells themselves. And so we'll probably learn of other effects as well.
I'm sure we would have fun discussing this matter was your father still alive. Let me ask you a final question about the class effect concept and its translation into practice. So why should a physician choose a compound within a class if it shows less certain benefits on important clinical outcomes? And to what extent can meta-analysis help?
Well, there, unfortunately, part of the answer is what we're allowed to use that is out of our control sometimes, that which compounds within a class are reimbursed often dictates. And so knowing broadly, does all the data look consistent, that you would feel comfortable if that were told to you that you have to use this drug and not that one. And that happens to all of us with the oral anticoagulants and with this class, SGLT2s, et cetera. So, but then the other aspect of it is it a class effect, et cetera, is to not get trapped into controversy and then not use the class at all. Because you say, oh, well, we're not sure if it's this one or that one. So they're still figuring it out. So I'm just going to stick with the tried and true older treatments. And so in part, finding in a meta-analysis the broad similarities to say this class is a terrific class and we've got to try and use it in all the eligible patients with less worry about exactly which agent has been my takeaway.
I'm with you. No doubt this is a major advancement in cardiovascular medicine, perhaps one of the most impressive in the past 10 years. So thank you very much, Chris. Now back to Filippo.
Yeah, regarding this fascinating new class of drug, we are going to publish in the European Journal a post-hoc analysis of DAPA-CKD showing that the reduction in mortality is mainly driven by non-cardiovascular mortality, so increasing the complexity and the interest, and perhaps suggesting that there are off-targets effects that really we need to understand better. But Chris, now I have a question from the audience, from Felicita Andreotti. The question is, do you think that the shorter than planned follow-up in VERTIS may have influenced the results?
I think not, in that in the end it was actually slightly longer than some of the other trials. So we end up with, I want to say it's 2.9 as the median follow-up, and so it's about the same as the other studies. And then largely the findings are consistent. We certainly had enough endpoints there. Longer duration is more important for looking at the kidney endpoints, as it turns out, because progression of renal disease does take time. And so that is a component, certainly with the last set of drugs with sotagliflozin, there was much shorter than planned follow-up duration. And so the renal endpoints are almost impossible to see a difference because like one trial was an average of about a year or so. So duration is important, but I think for VERTIS CV, we were in the same range and slightly longer than some. So I don't think that was a component, but an excellent question. And thank you.
Thank you, Chris. Thank you, Chris, very much for your answer. And now back to the New England. I have one more question for John. Well, you have, John, we can reveal this. You have a long-lasting friendship with the European Society of Cardiology and the European Heart Journal. Do you see ways to further strengthen this friendship?
Well, thank you, Filippo, for this question. Certainly, it has been my privilege to work with both the ESC and the European Heart Journal to try to find areas in which we can collaborate. I've been honored to serve as a co-chair for one session each year at the annual ESC Congress, although this, of course, was not possible the past year because of the more limited schedule due to the pandemic. And I should also mention that your predecessor, Professor Thomas Lucier, was kind enough to include me in an editors forum during the Paris meeting in 2019 that was arranged to discuss some of the changes taking place in the world of medical publishing, such as, for example, the development of Plan S, about which you and I have also had discussions, That forum struck me as a potentially very useful opportunity for sharing ideas and for supporting one another in our work. And I would be interested in the possibility of making such a forum, a regular event in the future.
Thank you, John. I definitely will work on that. And I have one final question for you. What are your suggestions for the future of EHJ Dialogues?
Well, let me say, first of all, that I've had the privilege of listening to some of the previous dialogues, and I've enjoyed them very much. It seems to me that you've created a sort of formula for these dialogues, combining what I might call personality and substance. That is, your guests have typically been leading figures in the field of cardiovascular medicine. So there's always going to be interest in hearing the views of major contributors to the field. But of course, sometimes conversations with celebrities are not always very substantive, and this can be a common issue. And you have avoided this trap in the dialogues by making sure that the discussions are indeed substantive and preparing sensitive questions. I think have responded with thoughtful and informative comments. So certainly, I would encourage you to continue the approach. You will know as well as I who some of the most appropriate leaders are and and what topics they might be best qualified to discuss. I do think that if you have not yet planned to include a dialogue on cardiovascular medicine in the COVID-19 pandemic, this would be a topic of current importance to your listeners, and something that I think, frankly, none of us feel we get enough information to fully inform us. So, any dialogue with somebody who has some experience that's worth relating would be of value in that area, I think.
Thank you, John. And this is indeed in my plans for future dialogues. Well, this concludes our dialogue today. And really, I wish to thank Chris Cannon, John Jarcho, and Carlo Patrono, the author, the editor, and the editorialist for this pleasant Socratic conversation. And I'm happy to announce that the next dialogue will be, as usual, the last Thursday of the month will be on March 25th at 17 Central European Time. Thank you again for your brilliant participation to this dialogue. Bye-bye.
Thank you very much. This was wonderful. Thanks to the audience too. Great to see 100 people listening in live. So that's wonderful. So long.
Bye, Chris. Bye, John.
Bye-bye, Carlo.